Thank you for joining us today for our webinar. Biomarkers are important in the clinic for, in part, understanding the progress of disease. In this presentation, we're going to hear about the use of our C-Series imaging system in biomarker research, focusing on a rare genetic disease, Niemann-Pick type C, or NPC. This webinar is hosted by Azure Biosystems. We are a company that develops products for protein and genomic research, from reagents to instrumentation to software. Azure Biosystems is based in California, near Silicon Valley, and continues producing in the USA. Our products are used by scientists that span many different fields of research, and we are proud to have employees supporting our customers in labs across the globe. Our speaker today is Dr. Stephanie Cologna. She earned her PhD at Texas A&M University and did her postdoctoral training at the National Institutes of Health. She is currently an assistant professor at the University of Illinois at Chicago. Stephanie, please go ahead.
Thank you, Lisa. I'm really excited to share with you today the work of my research laboratory, which is focused on differential proteomic and lipidomic signatures of the lysosomal storage disease, Niemann-Pick type C. When we think about neurodegeneration, we often think about the loss of cognitive and central nervous system abilities associated with aging. In this cartoon, we can consider that neurodegeneration is similar to the loss of leaves on a tree as the seasons change over time. However, unlike the ability of nature to regenerate the leaves on the tree each spring, in neurodegeneration, the loss of the central nervous system function is permanent. In my laboratory, we're interested in understanding what the chemical signatures are that change during neurodegeneration. And if we take that a step further, we can think about how those changes can tell us more about disease and how we can use them to develop therapies in the future. The major tool that we use in my laboratory is the mass spectrometer. Mass spectrometers allow us to do a variety of measurements associated with biomolecules, and perhaps the most common in our society is screening at the airport. The mass spectrometer provides us the ability to get very accurate mass measurement of various molecules. So when we think about neurodegenerative disorders as a whole, we think about how we have this progressive neuron loss, similar to the analogy of the leaves on the tree. We have degeneration of the central nervous system. And the most common of these disorders is typically read about includes Alzheimer's, Parkinson, or Huntington disease. However, there's a collection of genetic disorders termed lysosomal storage disorders, while individually rare, collectively are quite common. Many of these genetic disorders have central nervous system defects, including neurodegeneration. In neurodegenerative disorders, oftentimes we may observe misfolded proteins or fibrillation, and deposits are observed. However, this is not the case for all. Cholesterol is a molecule that's very important for cellular maintenance. Although it perhaps gets a bad rap in the media, in fact, it is synthesized enzymatically in mammals or obtained from the diet peripherally, and is very important for maintaining a variety of cellular functions. In the brain, cholesterol is synthesized endogenously and is tightly regulated. It's required for cell membrane structure and function. It acts as a signaling molecule. And cholesterol has been implicated in many different neurodegenerative disorders. So if we take these two items together, this idea of neurodegeneration and this idea of cholesterol and the potential for cholesterol dysfunction in neurodegeneration, I'd like to introduce to you a fatal neurodegenerative disorder called Niemann-Pick type C. So Niemann-Pick type C, or NPC, is autosomal recessive neurodegenerative disorder. It is biochemically classified based on accumulation of cholesterol and glycosphingolipids in the lysosomes. And it arises due to mutations of either the NPC1 or the NPC2 gene. Below, I'm showing a series of images of fibroblasts that have been collected from a control unaffected individual and 3 different NPC1 patients. You can appreciate the strong fluorescent signal, which represents the accumulation of unesterified cholesterol in lysosomal compartments. Now, why does this occur? In normal functioning cells, lipoproteins are taken up through receptor-mediated process, and cholesterol esters are delivered into the lysosome. These cholesterol esters are bound to the NPC2 protein, which is a small soluble lysosomal protein. An acid lipase can facilitate the cleavage of the fatty acid off of the cholesterol ester. The free unesterified cholesterol is then transferred between the NPC2 protein and the NPC1 protein. And then the NPC1 protein can then facilitate egress of cholesterol out of the lysosome back to the ER for recycling. You can imagine then that in the absence of functional NPC2 or NPC1 protein, that this recycling of cholesterol is impaired, and therefore cholesterol would accumulate within the lysosome, as I showed on the previous slide. As a consequence of this cholesterol trafficking impairment, many different clinical phenotypes are present. For example, NPC patients typically present with an enlarged liver and spleen. There's progressive neuronal loss, particularly of cerebellar Purkinje neurons. These are large body neurons that are in the cerebellum, which is shown in the bottom image on the right. The red staining shows Purkinje neurons stained with a marker called calbendin D. And on the top is a unaffected control animal, and on the bottom is a animal that lacks the NPC1 protein. You can appreciate the loss of the strong red signal and progressive degeneration from a rostral to caudal manner. The neuron degeneration mechanism in NPC is unclear, and there is some pathological overlap with Alzheimer's disease. However, they are distinctly different disorders. Furthermore, there's a large variation with respect to the age of onset and severity in NPC. As I mentioned, there's peripheral disease of the liver and spleen. And in the central nervous system, symptoms include ataxia or wobbliness, progressive dementia, seizures, and vertical gaze palsy. There is currently no FDA-approved therapy for NPC. However, a subset of patients do receive miglustat, which is approved for another lysosomal storage disease called Gaucher. The NPC team is currently involved in a clinical trial of 2-hydroxypropyl beta-cyclodextrin, which is shown on the right. Okay, so moving forward to the work that's done in my laboratory, we utilize the NPC1 null mouse model to do a variety of preclinical work. The NPC1 mouse model recapitulates the human disease quite well. At ages of 1 to 4 weeks, mutant animals are pre-symptomatic and look similar to their wild-type litter mates. They develop clinical symptoms such as tremors at about 5 to 6 weeks of age, ataxia at about 7 to 8 weeks of age, and are terminal by 3 months of age. So in this talk, what I'd like to share with you are a couple of studies that we've been carrying out using mass spectrometry as our primary tool to identify both protein and lipid biomarkers in this mouse model. I'll first share with you our efforts to look at liver proteomics and differential protein biomarkers, followed by an analysis of fatty acids, and I'll conclude with some comments on how we're trying to take this work and translate it into a more clinical setting. All right, so to get everybody on the same page with how we perform mass spectrometry-based proteomics in our laboratory, we rely on this the platform of bottom-up proteomics. In this experiment, proteins, whether they're isolated as single proteins or a collection from a tissue homogenate, for example, are first digested into peptides. These peptides then are typically separated using chromatographic means, and the accurate mass of each individual peptide that can be detected is recorded as shown in this spectrum. In addition to the accurate mass of the peptide, We're also able to perform tandem mass spectrometry, which allows us to sequence these individual peptide species. We perform this experiment iteratively thousands and thousands of times, and with bioinformatic tools are able to ultimately obtain peptide sequences, which then can be matched to genome databases to obtain protein identification. Depending on the experimental design, we can also allow for relative or absolute quantification of the proteins in our study. So using this bottom-up proteomic strategy, we first performed a differential analysis of liver proteins in the late stage of the null and PC1 mouse model that I showed you before. So we took liver from wild type and mutant animals, and we processed them identically, as shown here as A and B, and then performed that bottom-up proteomic sample preparation strategy along with chromatography and mass spectrometry to get our protein identifications. For quantification, we were able to take the peptide intensity for the same peptide in two different states, in this case, the control and the disease model, and allow us to get a relative fold change between control and disease. So by doing this in our tissue lysates, we then can take a list of our differential proteins and plot them in this volcano plot shown on the left. By using a fold change and a p-value cutoff, we can then show that a whole series of proteins are downregulated as well as some are upregulated in the NPC1 null mouse model. Furthermore, if we use these data sets to do a principal component analysis, we very clearly have separations between the wild type data sets and the mutants. So we further wanted to look into these proteins that were differentially expressed in our animal model. We had previously shown and observed in this analysis, of course, the loss of the NPC1 protein, and we therefore sought to confirm this by Western blot analysis. So as one would expect, we see no NPC1 protein in this knockout mouse model. We did observe an increase in our data set of fatty acid binding protein 5 and of fatty acid binding protein 7. And again, our Western blot analysis confirmed our mass spectrometry data set. The graphical presentation is shown below. So with this information, we were confident that our differential proteomics platform was working, and we wanted to investigate deeper some of the differences that we observed. So the first thing that we did was we performed KEGG pathway enrichment of the differential proteins in the NPC1 mouse model. Proteins that are associated with phagosome and lysosome and peroxisome were identified very highly with regard to the pathway enrichment. Other pathways include steroid hormone biosynthesis, oxidative phosphorylation, and so forth. We also evaluated how the differential proteins were distributed by cellular location. Many of the differential proteins were associated with the membrane or organelles, some with the cytoplasm and vesicles, for example. Now, these findings are quite broad, and so we then looked into our data a little bit further, and we performed a Western blot validation of our mass spectrometry data on 2 additional proteins. The top shows a progressive analysis of the liver from 3 weeks, so the asymptomatic time point in this animal model, to the more symptomatic and pre-terminal time points. For the lysosomal membrane protein LIMP2, which is increased at all of the time points analyzed, as well as our late stage proteomics analysis. And additionally, we looked at RAB7A, which is associated with lysosomes and autolysosomes. In this case, we do not see much of a difference at the early time points, but start to see a trend at 5 weeks and 7 weeks, and statistically significant alterations at 9 weeks of age. So while these individual proteins are quite interesting and provide us a means to look at further function, we also noticed something else about our data set. And looking at the proteins that were altered, I mentioned earlier that we saw an elevation of fatty acid binding protein 5 and fatty acid binding protein 7 in the liver tissue. Looking at additional proteins in that family, we also see a decrease of fatty acid binding protein 2. Furthermore, we started to evaluate additional proteins that were altered, which included a subset of proteins that are involved in the metabolism of omega-6 and omega-3 fatty acids. So this slide is a little bit busy, but I think it summarizes a lot of the data that we are collected. First, what I'd like to point out is that if we consider omega-6 and omega-3 fatty acids, which are considered dietary polyunsaturated fatty acids, they can then be metabolized through a variety of steps, which also includes transfer of these substrates across the blood-brain barrier. In red denotes all of the proteins that we observe to be decreased in our proteomics data set. And you'll notice that some of these enzymes have dual function across the two different pathways. Furthermore, in a separate study, we also looked at differential proteins in the cerebellum and the cerebral cortex of this mouse model. And in fact, we find elevation of these fatty acid binding proteins in these studies. So it's somewhat consistent with our liver findings. Interestingly, these proteins are responsible for transporting these fatty acids across the blood-brain barrier. So to investigate further, we wanted to first perform Western blot analysis as an orthogonal means to confirm the differential expression of these enzymes. But we also sought to look at these fatty acids individually. So I will show you a subset of these Western blots, and then note that all of the fatty acids shown with the stars, we also performed an additional mass spectrometry analysis. So that's shown here. For example, alpha-linoleic acid we found was decreased in the control, which is shown in black, versus the mutant, which is in white. This was also observed for arachidonic acid and oleic acid, among others. So looking at the free fatty acids, consistent with what our proteomics study showed us, was that we have a decrease of many of these free fatty acids in the liver tissue of the diseased animal model. These fatty acids can also be incorporated into other lipids, and so we considered the fact that incorporation could also be altered. In this case, we saw less significant changes between the two groups for many of the different lipids, but in fact still saw a loss of incorporation of arachidonic acid and some elevation of oleic acid in the mutant animals. So we can ask this question about what about other lipids? So another aspect of my research program has been to focus on lipids both in discovery and targeted based methods. In addition to standard liquid chromatography mass spectrometry methods, we also like to incorporate the ability to do spatial location of lipids. And to do this, we use mass spectrometry imaging. In mass spectrometry imaging, a tissue or other sample that you want to analyze, you can cryosection directly onto a target that's shown here. You can perform some sample preparation steps, and ultimately we use MALDI mass spectrometry, which requires a small organic matrix to be applied to the surface to facilitate ionization. Once the plate is ready, a laser can then be used to raster across the tissue of interest, in which each point of rastering is a pixel in which a mass spectrum is generated. And you can do this across the entire sample surface. Finally, you can get a two-dimensional false color plot of an analyte of interest based on mass. For example, this tall peak here, you might want to know where it's distributed across the entire tissue, and that's shown at the top. Whereas one of the medium intensity images might have a profile such as this. The beauty of this technology is the ability to spatially look at the distribution of biomolecules without the need for a tag. This is particularly important for lipid imaging because of the limited number of antibodies that are available to perform traditional staining or imaging modalities. So using this strategy, we also considered how other lipids might be changing in this disease. And in fact, we actually focus on the brain aspect of this disorder with our imaging studies. I'm showing here a mass spectrometry imaging analysis over a time course of disease progression in the same mouse model. So here we have 3 weeks, which is an asymptomatic time point, 7 weeks, which is a symptomatic, and 9 weeks, which is pre-terminal. So this follows the progressive neuronal death. And I'm showing a variety of different lipids, in this case, gangliosides. So GM3, GM2, and we have GM2 with 2 different fatty acid chains here. Interesting, at the 3-week time points, we see not much difference in the GM3, maybe a slight elevation in the wild type animal. However, for GM2, we see strong elevation of these 2 lipids in the mutant animal. Similarly, as we go to 7 weeks of age, much of the signal is lost for the wild type animals for GM2, and the elevation is observed in the mutant animals, with a strong amount of signal corresponding to lobule 10 in the cerebellum. And this is maintained at the 9 week of age. For GM3 on the top, this elevation starts to become more pronounced at 7 weeks of age, but you can appreciate it in lobule 10 more at the 9 week of age time point. Now, gangliosides have been reported to be altered in NPC before, and in fact have used antibody-based staining. One of the beauties, as I mentioned, is by doing mass-based imaging, we can differentiate the 2 different chain lengths because they have significantly different masses, in this case 1382.8 and 1410.8 for the 2 GM2 fatty acid chains. We've also looked at a variety of other lipids in NPC. This includes ceramides, which appear to be decreased or similar in the 3 weeks of time point. However, interestingly, as I mentioned, we have this progressive cerebellar degeneration. We start to see elevation of ceramides in the frontal lobules, in lobules 1 through 5, for example, at the 7 weeks of age. And at the 9 weeks of age, we see that signal kind of transfer to the back part of the cerebellum towards the later lobules. We don't see as much of a pattern for the hex ceramide. However, we do see kind of some remaining signals in the 9 week later cerebellar lobules. And we have some other species that we've mapped that are shown below. So ultimately, what can we do with this proteomic and lipid data that we're collecting in our laboratory? So we have now performed a number of differential proteomic and lipidomic studies, and we're interested in taking some of these candidate markers and testing them in patients. This may mean looking at blood or in cerebral spinal fluid. We're actively trying to understand the mechanisms contributing to the disease pathology. So these pathways that are altered and these markers that we've observed, how they may contribute to what we see clinically and what we know is defective in the disease. And ultimately, we'd like to be able to work with our collaborators to be able to develop drug and therapeutic options using this information. Whether these proteins and lipids are observed to change with the induction of drug therapy, or if they're actually targets that can be have drugs developed towards them. So in summary, I'd like to kind of tell you a little bit about what my lab has done. So we've used both discovery and targeted based protein and lipid biomarker analysis. I've shared with you some work in our liver and our brain model. We're trying to understand how biomarkers are present in cerebral spinal fluid and blood, as I mentioned, being able to translate these animal studies into humans, and eventually being able to evaluate these changes and other changes with experimental therapeutics that are being tested clinically. So in summary, hopefully I've introduced to you that NPC1 is a fatal neurodegenerative disorder without an approved therapy. We've integrated both proteomic and lipidomic studies with focus on liver and some on the brain. And we've performed mass spectrometry imaging to map molecules across surfaces. And biomarker studies are important because they can be investigated in patient specimens and inform clinicians in future work. If you're interested in learning more about NPC, Azure Biosystems, or mass spectrometry, these resources have been provided. And with that, I'd like to acknowledge those that helped us with this work, including the members of my laboratory, our collaborators, and a special thank you to the Era Parsidia Medical Research Foundation and Azure Biosystems. Thank you.
Thank you, Stephanie, for sharing this interesting work with us and for making Azure Imaging Systems part of your research. The Azure Imaging technology uses digital imaging to replace traditional film for Western blot. For any work that requires normalization and quantitation of Western blot bands, like you saw in this presentation, it's important to move to digital technology. The ability to confidently quantitate your bands is the true value of digital imaging. On film, chemiluminescent signals are easily saturated because of the small dynamic range of film, and this prevents accurate quantitative analysis. Digital imaging has a wide dynamic range, and the software can detect and report on saturated bands, letting you know which bands shouldn't be analyzed because their signal isn't linear to the amount on the blot. Additionally, the ability to take an image of a chemiluminescent sample and a color molecular weight marker is unique to digital imaging. Easily seeing the latter helps with post-image analysis. Azure Biosystems offers a wide range of digital imaging solutions with abilities to do chemiluminescent Westerns, fluorescent Westerns, gel documentation, and more. Learn more about our products, reagents, and workflows at azurebiosystems.com. We wish you great results with your Western blots. Thank you.